We're going to talk about developing draft guidance. And it's been an interesting process, and why we started, or why we embarked on the process of developing draft guidance has to do, I think I mentioned this morning, at the EMA released draft guidance in spring. But it's really more than that, more than just a copycat effort to the EMA. We are at a pivotal point in Duchenne, and there are issues that have to be addressed and considered and thoughtful. And we have been waiting a long time as patients for treatments, too long for most of us. So we wanted and interacted with the FDA on a regular basis, and in a conversation meeting with the agency last summer, we talked about our benefit risk, and we also talked about the EMA guidance and said we felt it was time that that guidance existed from the FDA. And as I mentioned this morning, Janet Woodcock said, actually, that's probably not going to happen. And with that, she suggested that we write the guidance. That was sort of, we decided we would begin by doing a similar policy form that was done in London in reference to the draft guidance of the EMA. And so we started on December 6th, and we had a policy forum in which a great number, 20 FDA representatives attended. There were 4 different groups. We were talking about unmet need. We were talking about the issues of biomarkers and surrogates. We were talking about these developing treatments for Duchenne. And so that was sort of the start. And then we drafted, and really drafted, and Dr. Charnos will probably tell you we twisted his arm around his back and wouldn't let him leave the room. And that's sort of the similar behavior we did to everyone sitting here on the steering committee. So the steering committee then, in various discussions, identified that there would be 7 working groups. And the working groups would be benefit risk, diagnosis, natural history, biomarkers, and we separated the biomarkers into biomarkers 1, which has to do with protein replacement, either utrophin or dystrophin, biomarkers 2, which was MRI, serum, and urine biomarkers, then clinical trial design and imperatives. And we first called that working group imperatives policy, but then we decided that it really was about the voice of the patient. So that's what it was, the imperatives, what the patient community believes and thinks is important that the FDA know as they review these applications that are submitted to them for review. So how we'll start is I'd like Dr. Tim Franson to talk about the relevance of the guidance and the process of doing guidance and its importance. And then we'll ask each of the working groups to talk about their particular working group charge, how they approached it, what, if any, were particular sticking points or controversial issues, and how they managed and what their conclusions were. So we'll start with Dr. Tim Franson.
Pat, thanks very much. And I would have to say congratulations and thanks to all of you. With each of those work groups, there was an incredible commitment of individuals who invested time from the patient, the parent, the scientific communities, and this is a wonderful collaboration. As you think about guidance, so what's the big deal? Think of it like missiles. Missiles have a guidance system. How do we direct all of our energies to something that can have an impact, that can compel change? Guidance is a process at FDA whereby a development roadmap for the FDA and sponsors' use is created. So if you will, it creates a pathway that allows for consistency and accountability and a level playing field for all the parties involved. This is very exciting, and it's even more exciting to me having observed FDA doing this over the years, and frequently it will take several years for a single disease-specific guidance to come forward. In this case, obviously, dedicated efforts of over 100 people in a 6-month period yielded exactly what PPMD and the group had promised to deliver to FDA. And this was, of course, informed by listening to what FDA had said about what would be helpful to them at the last December forum. So I really applaud that process, and I think this now creates a foundation for shared goals, and it should expedite the development and review of compounds and therefore speed things to patients. And I also would commend Holly and John Bridges and the others who worked on the Benefit Risk Foundation which is an important part of this guidance, and, if you will, is the real springboard to convincing FDA to look at Duchenne and other rare diseases differently. So this is a model. A patient group has never submitted guidance. In fact, groups were not even invited to do this until 2011, and we are the first. So what a thrill. Again, my hat's off to folks, and to me it involved folks laying out the ABCs. A for assertive, B for bridging, and C to compelling change. So my congrats to all up here, and thanks.
Thanks, Tim. Would you make just one comment on the fact that the steering committee decided there was a question of what's the shelf life of this document? How long is it going to last, and is it going to be submitted and then outdated? So could you comment on the thought or concern about a shelf life for this document?
That was certainly a concern, Pat, and very appropriately raised. It's our hope that as approvals come forward, we'll keep learning more and more from each other across companies, and therefore a guidance may indeed have a limited shelf life in some regards. But once you have a foundation, you have an ability to make segmental changes and to continue to build on this. And we had talked about, so if the guidance is good for 2 to 5 years, that's very good. And we hope to build on it from there.
And how will we update it? Is there a process involved? You want to speak to that?
In general, once FDA adopts whatever they choose to do, and we would anticipate a majority of this will be adopted by FDA, then it will be up to the community to stimulate FDA to consider recommendations for change. They are on no explicit schedule, but I would guess if we haven't seen change by 5 years, this community will be rattling sabers to have that happen.
Thank you. So we're going to go to our first, working group, and Holly PA chaired that with Dr. John Bridges. So I'm going to ask Holly to comment on benefit risk, any concerns or issues that they had, and any sticking points.
So, all right, let me give you a little bit of background about what we set out to do in the benefit risk working group, and then a little bit about the process we used and about our outcomes. All right. Hopefully that's better. Okay, so the benefit risk working group had really two major approaches. One of them was to focus on the ability to systematically collect and quantify people's preferences about treatments. And that's important because of a couple of reasons. So number one is the FDA deals with data, and we wanted to give them data that could inform what they were doing. It's the language they're used to. And the second reason is that testimony is really great and really powerful, but not everyone can get in front of the FDA. So we wanted to make sure that lots of people had their voice heard in the process. So those were very important. Another thing that we wanted to really focus on is making a case to the FDA of why Duchenne is different than some other disorders. So there's this feeling that in some disorders, the FDA, as a panel of experts, can make a judgment call about benefit at risk, the way kind of most people would think about it, but that the FDA really can't put themselves in your shoes, and they can't make a judgment call the way you all would because they haven't lived the experience that you've had. So we tried to make a case that the situation of a progressive disorder like Duchenne is different than many other disorders, and that benefit risk assessment by and for the community is really needed in a way that's not necessary in some other situations or as necessary. So the process that we use, you just want a little process. Okay, so there aren't any experts in benefit risk in Duchenne. So we had a little bit of a different situation than some of the other working groups where there are lots of experts in endpoints in Duchenne and treatment and management in Duchenne, but there really aren't any experts in benefit risk in Duchenne. So we got to bring together a very interesting group of people who do these health economic models. And it's in, there are probably none of them in the room, so I can tell you it's an interesting, different breed of researcher who are very focused on different things than some of us tend to be focused on. But it was really exciting to bring together these health economic folks from industry. So we had several people from very large biopharmaceutical companies that do this economic modeling to support their development of drugs. we were able to bring together folks in academic situations who do this sort of thing in usually schools of public health, as well as some clinician experts. And then, of course, we had parents and individuals with Duchenne muscular dystrophy who are the real experts at benefit risk in Duchenne. So the process, frankly, was a little bit bumpy at first as we all tried to speak a common language, and health economics has its own language. And those people don't know the Duchenne language. So we had a little bit of a culture clash in the beginning. But I think we got better as our situation progressed about being able to speak a similar language and make sure that we're defining terms and understanding each other. But those first couple of calls were a little bit tough. So the process was very collaborative. It became more and more collaborative about identifying shared goals. And in the end, the guidance part that we developed really is about making a case for the need to do these sorts of studies for disorders like Duchenne. There's a section on how those studies should be done, so some of the scientific information about the kind of methods that should be used, which is something the FDA is quite unfamiliar with, and they're trying to get themselves up to speed. There's, again, a focus on the importance of collecting this data instead of just doing patient testimony, so that they need to complement each other. And we also have information in there about the data we've collected, the pilot data, showing that there's risk tolerance for particular levels of benefit, but encouraging the FDA and encouraging sponsors to continue to collect this information over time, because people's preferences and treatments will change as more treatments become available and as things change in our community. So we talked about the need for that continual connection back to the community as these drugs are, as clinical trials are planned, and then as people put forward information to the FDA, that this needs to be a continual conversation with the community. I think those are the high points.
Thanks, Sally. So Kevin Flanagan was chair of the working group on diagnosis. Kevin?
So our remit was actually, or I should say our task was relatively easy in that diagnostics has certainly changed since 10 years. Over 10 years, we really surveyed the landscape first of, well, I should begin by saying we wanted to address the diagnostic odyssey was something we included in our discussions, letting the FDA know about the challenges of early recognition, highlighting the recommendations of the American Academy of Pediatrics for testing CK in all children with developmental or cognitive or motor delay, and hoping to shorten the diagnostic odyssey for parents. We addressed molecular diagnostics at the genetic level. That's we recognizing that the technologies are changing now and have been changing over the past few years and will continue to change, but suggesting some basic framework for what we would consider to be complete genetic testing to be performed in the future. That is, all exons interrogated, for example, in the setting of exon skipping or similar therapies. We we discussed as well protein-based diagnostics, so leaving aside the question of dystrophin as a pharmacodynamic biomarker or disease progression biomarker, leaving that for the biomarker section. We offered guidance on sort of the standard clinical diagnostics of dystrophin expression on a standard biopsy, and did mention, of course, that this was a different scenario than the very specialized fold changes that one has to demonstrate in a clinical trial. And then touched a little bit on the fact that in several points touched on the fact that genetic mutations by themselves do not define phenotype, that there are abundant examples of exceptions to the predicted genotype, so that the need for clinical acumen as well in the diagnosis of the disease. So really, I think that covers, we had a really relatively discrete section, and I think that covers it.
Thanks, Kevin. And then we'll go to natural history. Dr. Craig McDonald chaired the working group on natural history.
Thanks, Pat. So the natural history group really, I think, focused on the importance of really having sponsors being aware of how the current understanding of natural history really provides opportunities for evaluation of new treatments across the entire spectrum of disease. So the emphasis here was really focusing on the entire spectrum of Duchenne dystrophy. Much of this work originated by the work dating back to 2010. There was an international DMD clinical outcomes working group that has been meeting on almost an annual basis. Many of those participants are here at this meeting. People like Professor Natalie Gomans from Belgium, Professor Eugenio Mercuri from Italy. We've also had Katie Bushby from the UK, Doug Bigger from Canada, and we had industry representatives in this group, as well as parent participation in this working group as well. I think that we focused on initially the importance of really looking at endpoints with regard to their properties in terms of reliability and validity, the relationship of endpoints with other validated endpoints, whether there was data available on endpoints, that normative data that was available, whether there was natural history data available in Duchenne populations. In addition, there was an emphasis on whether there was evidence of responsiveness of the endpoints to given therapeutic interventions with Duchenne dystrophy. And then finally, whether there was data on clinical meaningfulness, and then whether the endpoints had some experience with regard to implementation in multi-center natural history trials. We went ahead, actually taking the lead of the care guidelines. We defined the stages of Duchenne dystrophy and really focused, I think, again, on all stages of the disease, neonates, infancy, young children, ages 1 to 42 months, so the early ambulant, also the young ambulatory from ages 4 to 7, late ambulatory patients from 7 to 13 years of age, early non-ambulatory as well as late non-ambulatory as well. And then we also focused on data that's available with regard to clinically meaningful milestones, looking at ambulatory functions and milestones, because many of our endpoints actually predict the loss of clinically meaningful milestones. And we also defined non-ambulatory milestones as well, such as ability to reach over the head, or the ability to self-feed without adaptations, ability to place the hand on the tabletop, ability to use a computer, distal functions. We also focused on pulmonary milestones. The care guidelines have actually given us parameters to focus on, which actually should alert the clinical team to actually implement different levels of surveillance and different pulmonary interventions. So there could be milestones such as 40% forced vital capacity predicted values, or 30% predicted forced vital capacity, or particular parameters on maximal expiratory pressure, which actually have, in the actual pulmonary guidelines that have been published by the American Thoracic Society, as well as the Duchenne care guidelines, actually have been deemed to be clinically meaningful in terms of a clinician and management team actually implementing different management strategies. And then I think there's also cardiac milestones, which we touched on as well. And then we looked at issues as to how optimal medical management really affects the course of Duchenne dystrophy. And I think this has been important in some of the recent trials, multi-center trials that have been done, where sites have been utilized that perhaps haven't had experience participating in Duchenne clinical trials and may actually provide a very different standard of care. So we thought this was important to focus on. But it was, I think it was with the recognition that even with current care standards, there is still, I think, a sense of urgency in terms of treating this disease. For instance, in the prospective Synergy Natural History Study of 340 children with Duchenne, there are still children, there's been a large number of children that have died during that prospective follow-up. and really at very young ages as well as older ages. So even though we have interventions now that really can have a tremendous impact on lifespan and natural history of disease, there's still a sense of urgency in treating Duchenne that I think the regulatory authorities need to consider and need to be aware of. We focused on glucocorticoid therapy, on contracture management, spine deformity management, pulmonary management, cardiac management, as really important issues that need to be considered in terms of clinical trial design. And then finally, we focused on issues of heterogeneity of Duchenne disease progression, looking at important issues such as disease severity, stages of disease, and baseline function, age of the patient participating, and then issues such as genetic predictors of disease progression, the mutation-specific genetic predictors, as well as genetic modifiers that Dr. Flanagan and others around the world have been focusing on. Some of the recent genetic polymorphisms, such as the LTBP4 polymorphism and the osteopontin polymorphism, which has actually been shown to be actually more of an issue modifying patients' response to corticosteroid management than actual inherent disease progression. And then we looked at issues of night splinting, physical therapy, and other standard interventions. And then finally, we finished with some comments focusing on just the rigor with natural history data collection that's needed to satisfy FDA requirements. And I think this was a good transition to the next section on clinical trial designs, outcome measures, and considerations that Dr. Lawrence Charnas chaired this session.
So, Dr. Charnas, do you want to talk about your trial design?
Sure. Am I on?
I think so.
Yes? No. Can you hear me?
Yes.
Okay. The working group 6 was clinical trial design, endpoints, and consideration. And the history of this working group was interesting. In the beginning, it was going to be separated between trial design and endpoints. And then, as you heard from Dr. McDonald, you can't really talk about those things unless you understand natural history, and he was focusing on endpoints. So it became more on how do you design trials and what are the special considerations for Duchenne. And so the first thing we had to do is recognize we didn't know what it was going to look like, and so we started out with a joint goal, and Dr. McDonald and I first agreed on what the content would be of this working group. The second aspect was to identify who were the members. And as you can imagine, there are a very large number of people who are gifted and could do this work. And so we created a list in the specific areas that they could contribute. And the list was about 36 to 40 people who could serve on this group. And in the spirit of timeliness and feasibility, we knew we couldn't possibly get people from Europe and the United States in 4 months to agree and meet and do things with 40 people. It just wasn't feasible. So we then used a technique that I don't believe PPMD can patent. It's called the stair technique, where you put the names of people on a piece of paper and you throw them down the stairs, and then you pre-select which steps are the people you're going to capture. And we did it because I couldn't think of a better way to select people. And so once the people fell on the lucky step, they then got an email from me inviting them to participate. I'm glad you're laughing. It means that I haven't put everybody to sleep. Then we got people together and tried to figure out how we were going to incorporate what was natural history and how we were going to apply that. And there was a fair bit of overlap between natural history and trial design, but it was not complete, and that worked out very well because we had insight into what was happening. And then we focused on what we thought would be very helpful for industry and sponsors and also for the community to understand what is unique about Duchenne that people may have missed. We have an explicit statement of the difficulty in a rare disease where individuals go through specific stages of diagnosis, eligibility in terms of performing specific tests, and then losing abilities but being able to do other things. And to my knowledge, there's no other disease in the rare disease spectrum that looks like this and has been so categorized. So that was an important concept to move from the natural history to spread that out to understand the populations and how trials might look to cover that. There are no instruments that are perfect for all subjects in a study and across the entire spectrum. So we had to call that out. And being forward-looking, we identified what the perfect instrument would look like. And we got feedback from industry saying, well, that's not how we do business. And absolutely true, that isn't. But that isn't necessarily the best place to be forever. Maybe there's a way to do it. Again, we don't know. And so at the end of the day, we have areas where there's good consensus. We know exactly what instruments do and how they behave. And then there are areas where we think we need instruments, but we don't really know exactly how to do it. And that is for future work and gives some guidance on what people need to do to go further. Thank you.
Thank you. Thank you. So then we're going to back up and go to the biomarkers. And I went from natural history to trial design because I thought it made sense. I hope you agree. And now we're going to back up and look at the biomarkers. As I mentioned, those were separated into two different groups. Biomarkers 1 was chaired by Justin Fallon, and that was focusing in on dystrophin and utrophin. Justin?
Okay, can you? Yes, you can. Okay, so it's my privilege to lead the biomarker group, taking over from John Porter. And basically, we were comprised of a very broad and very, very hardworking and knowledgeable composition in the working group that included people from industry, all the major players, academia, clinicians who treat boys and also run trials. And another feature of this was that this was an international effort. And so particularly, for example, Annemieke Arsma-Rus was on the committee, and she was involved intimately in the EMEA guidance. So we had we're working to coordinate internationally the voice that we're doing. These are independent documents, but we're in close communication. I think that's important as we go forward. And, of course, all the voices. So that was the composition of the committee. And we were on the biopsy-based biomarkers. And we thought a lot about this, as you do too, and we started out with a couple of questions. One is, why do you need a biopsy, and what's the value of it? You want to make sure, of course, that this is something that's important to the trial, to developing the therapy, and that it is worth the considerable hardship that the boys and the families need to go to. So in that line, how do you get the best possible, if you need a biopsy, how do you get the best possible one? How do you handle it? How do you make sure you get the best information? How do you make sure that the information that you get can be shared and rationalized with other groups doing similar work? So these were the drivers behind our thinking. And again, to get a biopsy, particularly early on in the development of a therapy, is very important because, as you heard this morning, it's important to know if the drug that you're testing is on target and is working by the mechanism. And so biopsies are one of the most specific ways and valuable ways to do that. So we worked a lot on best practices, so not only for people in this room or at this table, but also sponsors who may be coming in, hopefully, new people. And where do they go? And trying to say, here's where you look, here are the potential pitfalls, here's what you can do to mitigate those, and have that. So again, getting the most out of it. And then we were measuring. We spent time talking about dystrophin, about eutrophin, and about the associated proteins in terms of reassembling this complex that's anchored by these proteins at the muscle, and the compromising that complex, of course, is what leads to the damage. And we worked on primarily two methodologies for measuring. One is immunofluorescence. Another one is Western blot. These are very complementary methods. And we also talked about, and this I should say is all the work that we talked about was based on published work. Okay, that was a guideline. And then also we did flag some emerging technologies, since this is a forward-looking document, and among those was mass spectroscopy. And then the questions of how to best get consistency, reliability, reproducibility in these measures, and then get them disseminated. And we were very supported and aided by a publication just coming out. Kevin Flanagan played a major role in this, where an international group of laboratories in coordination, but each individually analyzed the same set of samples, both by immunofluorescence and Western blot, and got very concordant information. But the message there, that the way they did that was by coordinating up front, and to share the information, share the knowledge, share the methodologies. With that, we now have a lot of confidence that we can compare and that we can rely on the kind of information that's coming from the multiple sources. So that was our group.
Thank you. The biomarkers, too, was Lee Sweeney looking at MRI, serum, and urine biomarkers.
Yeah, so our charge was to review what has been done on non-invasive biomarkers, that is, biomarkers that don't involve biopsies, and that they fall into two general classes. One is the biomarkers that are accessible either from the blood or from the urine, and those are biomarkers that essentially integrate what's going on in all of the muscles in the body. So they don't tell you anything specifically about which group of muscles is the most affected, but they give you sort of a whole body readout on disease progression. The underlying premise of all these biomarkers, as you heard from Justin, is that they will inform early decisions in trials, so that one can tell if one's drug, for instance, is having the desired effect by looking for a response in these biomarkers. But the long-term goal is that hopefully many of these biomarkers would be robust enough, but that will only be proven when they're looked at in an exploratory manner in trials, robust enough to eventually be used as some sort of surrogate to speed the approval of drugs before you have enough longitudinal functional data to support the ultimate approval. So getting back to the serum and urine biomarkers, we reviewed both proteins, metabolic products, and RNA species that can be detected in the blood and urine. We essentially reviewed what has been done to date on those and some commentary on what we see the promise for the future around all of these different classes of biomarkers and the technologies that are being used to detect them, and what the pros and cons of those would be. Then the second half, which included MR, was a section on imaging modalities. MRI was the one that we spent the most time discussing because the most has been done with it, but obviously there are other imaging modalities such as DEXA and impedance mapping and a number of others that have been used to different, and echo, that have been used to different degrees, both in skeletal and in, certainly in the case of echo and MRIs, both in cardiac muscle, for cardiac muscle as well. So we discussed the pros and cons of those, the technology, the limitations, and again sort of laid out what we thought sponsors might, what might be useful to sponsors as far as what information they could get, how robust that information might be, and then ultimately encouraging the FDA to think about which of these biomarkers they might want to work with industry and academicians to eventually qualify as various levels of biomarkers. But as I said, the long-term goal is to get a surrogate imaging biomarker in place that would allow one to make early decisions in trials and eventually maybe even get early approval, accelerated approval in trials. So that was our charge, and as you can tell from what I'm saying, these biomarkers have yet to be proven. They really have to be looked at in trials with interventions so that we can see how well they would have predicted whatever that intervention is going to do in terms of the functional outcomes. And so they're a ways off, but it's important that we get them in the pipeline, developed, qualified, because ultimately I know you don't like the mini biopsies and you don't like the long length of the trials based on having to wait 1 or 2 years to see functional changes. So I think these biomarkers are going to be critically important as we move down the development path.
Thanks, Lee. And on the imperatives group, Neera and I were chairing that imperatives group, so I'm going to ask Neera to talk about the imperatives and how she included, and we included, the patient voice and the patient imperatives.
The patient imperatives were really the cornerstone of the whole document. The most important part of this is that the patient voice was to be heard. The unmet need, the urgency was primary, and the fact that we need an inclusion across a broad spectrum of the disease was critical, and to move away from placebo was extremely important as well. That we as a community are willing to accept risk. We live every day with risk, and that we want treatment for our boys.
All right, thank you. All right, so maybe Tim, or Kevin, or Craig, we talked about publishing the document, and I wondered if you are still on that track and what you've thought about, or if you've thought about that and where we're moving.
Yeah, I think we'd like to publish this in a peer-reviewed journal, preferably a journal that is read by the large neuromuscular community, and also we'd like to publish it in a journal that would give free public access to this. So there's a number of potential journals that we're having discussions with at this point in time, but we'd like to try to expeditiously get this published.
Thank you. So I wondered if you all had questions about the guidance. If there's questions about a specific area of the guidance that I don't know if you've had time to read the guidance, and if there are questions, please feel free to come to the mic and ask them, and to the degree that's possible, we will answer them. I think this guidance, to the real extent possible, really is a consensus by the community. And I'd like to know from others, if you felt, Justin, maybe you in particular, on the dystrophin issue, were you able, do you think, to arrive at some sort of general consensus about dystrophin and its biodynamic qualities, and whether or not and when or not it would become a surrogate?
We talked a lot about that, of course. And so the state of the field, so again, what we were looking at is the published data, and where are we? So in order to get a surrogate marker, you need to be able to measure the amount of dystrophin that's present. Currently, up until very recently, there's been no consensus about how to do that. That has now changed. And so there now is good consensus about how to measure. That was the most important thing. Now, with that foundation, and again, having this reproducibility and reliability, we're in a position to do the next stage where one can take that information and then correlate that with what is going to have clinical benefit. So it's not there yet, but it's absolutely on the path to it.
Good, thank you. And Dr. Charnas, about novel trial designs. In terms of Duchenne, what we would like is fewer patients need to be involved, and a quicker process and a shortened approval process. So in your discussions about trial designs, are you aware of, in discussions, have you come across designs that might be more amenable to take a look in a more expedited way in Duchenne?
So the major concerns relate to the ability to detect a change, and so that depends on the instrument that one measures change. There's broad agreement that accurate natural history studies could shrink the size of studies so that if one had to use a placebo, that it would be done in a smaller number of boys. If the extent of change could be quantitated more precisely, then the trial might be shorter, or one might be able to use a criteria for when to switch from placebo to treatment. But there's also, I must say, a large group of boys for which there really are not adequate outcome measures, which to me is the largest unmet need and where the field really needs to move. And we identified those and talked about ways that people might begin to look at the broader community and develop new ways to measure that.
And Craig, could you comment on some of those outcomes?
Yeah, I think with regard to the non-ambulatory population, there has been international efforts to standardize a measure of upper limb function that actually has a theoretical construct that corresponds to the progression of the disease in non-ambulatory populations, really going initially from the loss of shoulder functions to elbow functions to distal hand functions. I think that there's been some nice reproducibility that's been demonstrated, some rash analysis, meaning that we know now that a certain measure on that scale actually is truly less severe than another measure on that scale. So we're beginning to actually understand the scale itself, and that's the performance of upper limb measure. So I think there's been great strides that have been done. That scale has actually been implemented now into some of the extension trials, as well as the recent Lilly tadalafil trial. So I think that measure has really been rapidly employed. In addition, there are quantitative measures of reachable workspace that I think hold promise to actually get very sensitive measures of changes in upper limb function. In addition, there are some measures of distal hand function that the French have been evaluating that hold promise, but some of the instrumentation is quite expensive, and none of these methodologies have really been employed on a wide-scale basis in clinical trials or in multi-center natural history trials. And then I think there are international collaborative efforts underway to try to evaluate some of the item banks for the patient-reported outcome measures of health-related quality of life and other domains that are of clinically meaningful to patients and family members. So there's a lot of efforts underway with regard to that. I think in the infant population, I think there are more challenges. I think Dr. Charnas has a lot of experience with other genetic diseases and other neurodegenerative conditions in terms of the challenges of doing developmental-based testing and testing of functional milestones in infants and toddlers and young children. That being said, there have been efforts at least underway to collect natural history data on some of the developmental milestones in young children. And I think that that's at least being employed in this infant steroid trial. I think, Dr. Charnas, did you want to comment further about the challenge of young child endpoints?
Yeah. So there are standard ways to measure how a child is doing, and this is done routinely in all preschool evaluation. Unfortunately, there's no standard way once a child times out, reaches the maximum age of the instrument, to know how to move to the next instrument. And some of the instruments that are used are limited to the country in which they can be used, in which they're validated. Some of the instruments have not been translated into more than 2 or 3 languages. And some of this is as simple to say that if one uses an instrument in the United States, It typically has to be available in English and American Spanish. If we go to Spain or Mexico or Colombia or Venezuela or Argentina, the language is different, and each of these instruments has to be validated. And that creates a huge limitation on how you can do studies in rare diseases if you have to have country-specific tools. So it was that kind of detail that we tried to at least point out so that we understood and could compare apples to apples.
I want to just also mention, as another approach to endpoints that would be employed in young children, is obviously if you're taking a 2-year-old child, there may be some degree of challenge with regard to doing MRI imaging on them in terms of potential need for the child to sit still. Doing it perhaps at nighttime when they're asleep have been some approaches. But there's another measure of electrical impedance myography that has been worked on by Deanna Escalar and her colleagues. This has been employed in some Duchenne dystrophy multi-center investigations and is currently being employed in one of the Neuronext trials of spinal muscular atrophy in babies, healthy babies, as well as babies with spinal muscular atrophy. And this is essentially a measure of body composition that's done focally in a particular muscle, such as the biceps muscle or the quadriceps muscle in the leg, and so forth. And again, this is a measure that may hold some promise in younger children.
Thanks, Craig. Brian, you had a question?
Yes, let me begin by saying thanks to everybody who was involved in this effort. I think it brought a lot of people who have a great interest in the community together to create a wonderful template that I'm sure will be useful for clinical trial sponsors and people who want to understand more about Duchenne muscular dystrophy. One of the things I was unclear about is the time frame for updating the document. As has been mentioned, many of the outcome measures are under development or they're planned, and with the optimism that we can move away from some of the more invasive procedures. And what is the time frame for updating it and improving the utility of this for industry?
Thanks, Brian. So we did set standards at the beginning of this, the development of this document, and the standards were that information or data that was be included in the document would be published or accepted for publication by a certain date, and I believe it was the end of July. So we didn't want anything that was unpublished data or other, and so that's what's in the document currently. I don't suspect, and I'm going to ask Tim and others to comment on this, that the entire document will need updating in a short period of time, but rather as new knowledge comes about dystrophin, about outcomes, and perhaps about patient-reported outcomes, that subsets of the document would be able to be amended. And so, Tim, do you want to comment?
Sure. It's a great question, and I think that question comes up with multiple guidances. How often does one need to refresh? FDA and EMA do not have standard timeframes for the lifespan of such a document. I think Pat has hit the most important points, which are with each new application, there's going to be learning. As that learning accumulates, as a community, we all will need to be able to revisit on a regular basis every several years. Do we think the progress has collectively been enough to update a section or even more? So I'm not sure any of us can predict how much progress will occur, but if other therapy areas are a good guideline, once the initial 1, 2, 3 applications are approved, there becomes really a cascade of information, which is great.
So it was a very aggressive time frame for the development of the document, and I'm glad that a lot of time and effort was spent on making sure that everything was done correctly, and that was my understanding, was that certain elements could be updated as necessary, particularly outcome measures, as that there's there's approval for them, there's acceptance and utility for the community. Thank you.
Yes, thanks, Brian. Holly, just one question for you as well, and that is, as some of these new therapies come online, and are you planning, or do you suspect, as you investigate or ask the benefit-risk equation to the young adults with Duchenne, that perhaps the equation will change, or do you see any challenges there?
Is that okay? Yeah, it's on. So that's a really good question. So the way that the experiments are set up that we used and that we encourage use for drug development is that you would expect actually people's preferences to change. And those preferences may change because you're asking someone with Duchenne versus their parent, and that is our next step, actually starting this summer, to look at the preferences of individuals with Duchenne who are teens and adults. But it also might be because the person gets older and their preferences change because they have different priorities. Or it might be because, fingers crossed, we have a new treatment on the market in a year, and that makes people think differently about what their options are. So we do think that this is going to require ongoing communication with the community to understand how people's risk tolerance will vary and how their perception of benefit will vary. Right now, it appears that people will accept a relatively low level of benefit, but when we have a couple drugs on the market, people may ask for quite a lot more out of their drugs, and I think that would be reasonable. So we'll keep looking at it.
Thank you. And Lee, why can we or not rely on the serum CK to tell us something about disease progression?
Well, the problem that a lot of the blood biomarkers have is that they're dependent on the muscle mass and the activity that the muscles are undergoing. Because a lot of it's not true of all of the blood biomarkers, but for the majority of them, they're not released unless the muscle's being used and being actively damaged. And so that creates great variability depending on what the patient's been doing prior to in the period, the 2 days or so prior to when the blood is drawn. The other issue is, of course, you'd have to normalize for the muscle mass that the patient still has, and that can be done to some extent using DEXA, but that's not totally reliable in the sense that as different muscle groups are being lost, then the groups that are being preserved are obviously the ones that are not being damaged at the same rate. And so you're going to see the CK going down even per unit muscle mass just because the muscles that are involved, that are still intact in the disease, are changing. So this is why we feel that one has to have better imaging techniques such as MRI to directly look at the muscles of interest and really understand what their integrity is. CK and other blood biomarkers can give you short-term information. They can tell you whether something is responding to a drug over a fairly short time period, and give you some indication that your drug is doing something. But sometimes in our mice we see CKs go down because the drugs makes the animals immobile. They just sleep all the time, and so that makes the CK go down too. So it's a problematic biomarker. It's still a useful one, but it's by far not a perfect biomarker, and so we're seeking better blood biomarkers as well as better ways to do imaging.
Thank you. And Kevin, in a diagnosis, are there any indications or any times when genetic testing is insufficient and you require a muscle biopsy?
Yes, it is important to remember that as good as molecular diagnostics are for genomic DNA analysis. Genomic DNA, of course, is what we analyze out of white blood cells, out of a blood sample. There are about 5% of cases of what is classic Duchenne that, in fact, are undetectable by that method. These are people who have an unusual pattern of rearrangement of splicing. Generally, they're called pseudoexon mutations, and these are where a deep intronic mutation makes a false splice signal, and therefore you include some intronic material in the final gene message. Those are not detectable by any of our current standard diagnostic tests and are likely to remain undetectable for some time. So biopsy still has a place, absolutely. And somewhere, probably not quite, in somewhere less than 5% of patients it's required for diagnosis.
And one last question about biopsies, because they involve typically anesthesia, because we do open biopsies. Isn't there a way to do a needle biopsy so that you can have some local anesthesia and not so traumatic? And why is it different? I mean, in terms of specimen, I understand, but isn't there a way to image what you're looking, image the muscle and then take a specimen that's going to give you the best possible piece of tissue to examine?
There are very good centers that do needle biopsy. I would say that for most clinicians, at least most clinicians in the States, feel that the problem with needle biopsy is a relatively small specimen and needs to be oriented properly to do proper imaging and so on. Needle biopsies are great for doing a Western blot or doing a very small fragment of muscle. One of the things I should mention is for that 5% of patients, roughly speaking, 95% of things that look like Duchenne in a 5-year-old boy are Duchenne. But if you exclude those people who've had genetic diagnosis from blood, then the number of other diseases that could be mimics of Duchenne actually arise. So the reason for doing an open biopsy is not solely to analyze the dystrophin expression, but you then can exclude all sorts of other muscular dystrophies, like the severe childhood autosomal recessive versions of limb-girdle muscular dystrophies, or like FKRP-associated diseases. So one of the reasons to do an open biopsy is it leaves sufficient tissue for a variety of testing, both more detailed dystrophin studies, but then as well specialized mRNA studies or other things. That's not to say if your clinician someplace recommends needle biopsy or suggests needle biopsy, it can be done, but it should be done in those centers that do it routinely and make a practice of analyzing in that fashion.
Okay, thank you. And Neera, do you want to comment on, some of the things we talked about in terms of having us as patients understand and companies to pre-specify some of their plans? Do you want to talk about that?
So,
Compassion and equal to.
yes. With companies letting us know what their plans are and what our plans are, the compassionate use issue is something that we would all want, but the problem with compassionate use to a large extent is it's a real problem for the company, and I think it slows the field for everyone, for all our boys. And that's why actually increasing trials or making a larger number of trials and having trials across the disease spectrum really does help us increase exposure to the different possible therapeutics.
Thank you. So it's 1:20, and I'm getting red flags from Kimberly, so we're going to end this session. I first of all wanted to thank Teo Smart here in the audience. Teo, we've gotten to know Teo, and Mark Kruger and Associates were part of this guidance that aren't mentioned. Teo was the writer. MK&A was the project manager who, I guess we could say, herded cats. Really kept this project on track, and Teo, I think, did a magnificent job of really reflecting the patient voice and the guidance to date. And so I wanted to make you aware of Teo and his work with us. He at one point, I was quite certain he wasn't sleeping, and there was a day that I thought he was going to run away. But that said, he didn't, and I think as a community we should be proud of this guidance. To the greatest extent possible, 200 people came to consensus on this document, and 200 people with 200 opinions, and as I say, in this community there are considerable opinions, which is wonderful, but to bring that down to a single guidance that could be submitted is amazing. And the fact that FDA has now said they're going to issue their own guidance is really terrific. And I'm hopeful that the CAB, the Community Advisory Board, will stay together and continue to discuss when and if new information, new knowledge arises, what's important to them and what we need to think about. And the working groups, we already know the chairs, we already know how to bend their arm around their back and make them stay with us. And as things go forward, I'm hopeful that we'll adopt and adapt and move forward with the guidance in terms of new knowledge. So thank you all.